So good morning, everybody. I'll be talking about coronavirus disease 19 and digestive system. Before I go to that, I want to briefly tell you the coronavirus disease 19 epidemiology and then how it spreads so that we can actually explain the digestive issues later in a more clear way. We all know as of January 2020, only 4 countries were affected by coronavirus disease. Obviously, the country from where it originated is China, and that was affected, and a few adjoining countries. As of February 15, 2020, almost 20 countries were affected, and subsequently, as of March, 66 countries, and towards the later part of March, 5 more countries were added. And today we are talking about in September, and we all know almost every country in the world has been affected by coronavirus disease 19 pandemic. Now, how does the infection occur in human? Now, there are some issues which are hypothetical till now. I would say these are popularly believed hypotheses. People have wanted us to believe this. So this says that initially it originated from bat. Some theories suggest it might have originated from pangolin. But there are genomic studies which showed that the sequence of current SARS-CoV-2 virus, which is causing the pandemic in human, has a greater homology with pangolin virus than the bat virus. And people tried to make us believe that actually because in China, pangolin, bat, all these are eaten, and even these are eaten raw. So therefore, this led to coronavirus entering into human body. Pangolin, by the way, is a sort of animal which cannot be sacrificed, but obviously if somebody is eating pangolin meat, it is outside the law. Now what happens is if we believe this hypothesis, it means coronavirus disease initially entered human through GI tract. Now, subsequently, once the virus has entered the body, then there is a direct contact. So it is an aerosol transmission from person to person. Now, once this enters into human body, how does it enter into the cell? We know angiotensin converting enzyme 2 or ACE2 is the receptor to which the spike protein of the virus joins. Also, there is another receptor on the cell surface that is a TMPRSS receptor or transmembrane protein receptor through which also the virus enters. The virus undergoes endocytosis and then it produces an endosome, and inside the cell there is endosomal acidification. People have realized that this viral endosomal acidification occurs because the sialic acid of the membrane being released, and that lead to endosomal acidification so that the membrane of endosome is broken or dissolved and the virus RNA enters inside the cell. Now question is, once the virus RNA enters inside the cell, what does it do? This virus has a capacity to do cytokine storm. The predominant cytokine is interleukin 6. The much of the manifestation that the virus causes in our body is because of the liberation of the cytokine rather than due to the virus itself. So it is a virus which activates our immune system, liberates a lot of cytokine, causes cytokine storm, and then this leads to acute respiratory syndrome and systemic problem. This is one of the paper which was published in Frontier in Immunology, where they took mild to moderate coronavirus disease 19 patients, severe, critical, and those who died. and they looked at their total T cell, they looked at their CD8 T cell, they looked at their CD4 T cell, and they found that as you get more and more severe illness, there is a T cell exhaustion. That means there is so much stimulation to the T cell that they become exhausted, and this leads to T cell exhaustion. So it is a cytokine storm and immune activation which is responsible for the illness. Now, what is the situation of COVID-19 in India? We all know that COVID-19 for the first time in the country or India was reported in January 30, 2020. But in a short span of nearly 6 or 7 months, the country has seen its devastating epidemic. Almost every state has been affected. Now, this is a paper which was published in May 2020. Obviously, this is an old data, and it shows the distribution of the cases in the different states of the country, and you can clearly see that this is an epidemic involving the whole country. Now, what happens to the GI tract in COVID-19? If we believe the hypothesis that it is the pangolin meat and the bat meat which led to entry of this zoonotic virus into the human, then you have to believe that it enters through the GI tract. Now, why did it enter through the GI tract? This is because ACE2 receptor are in plenty in the GI tract. They are there in the small bowel, they are there in the colon, they are there in the esophagus. So because the ACE2 receptor is in plenty in the GI tract, that's why GI tract gets involved. You need proof for this. Here is the proof. Actually, this is a study which was published in JAMA, where they looked at young children and the older population, and they found young children get much less severe COVID-19 than the older population, and this is well known. But in addition, in this paper, they looked at ACE2 gene expression. So they found that ACE2 gene expression in the young children, nasopharyngeal epithelium, is very less compared to older people, and this may be the explanation why the disease is much less severe and infection is much less frequent among the younger population. Now, GI symptoms are common in COVID-19. There are now plenty of studies in the whole world. Much of the data came from China, but subsequently we have data from Europe, we have data from North America, and so on. Now, the GI symptoms include anorexia, nausea, vomiting, diarrhea, abdominal pain, and discomfort. And there are now meta-analysis on these. I will show you that 17% of the people with COVID-19 actually have one or more GI symptoms. This is an old meta-analysis of 60 studies, 4,243 patients, and they found a pooled prevalence of 17% people had gastrointestinal symptoms. And what they found is those who had severe COVID-19, 17% of them had gastrointestinal symptoms compared to mild COVID-19 or non-severe COVID-19, where 12% of them had GI symptoms, which means there is a trend towards higher frequency of GI symptoms in patients with severe COVID-19 than milder COVID-19, though this difference was not quite significant. There are two symptoms which are unique. Like you get people having common cold, and the common cold symptoms are very similar to COVID-19 symptoms. Like common cold, you have fever, you have loss of appetite, you have myalgia, you have arthralgia, you may have cough, nasal congestion, maybe irritation of the throat. These are all there in COVID-19 as well. But what happens is GI symptoms are uncommon in common cold usual type. Other two things that are uncommon are gustatory and olfactory dysfunction. That means anosmia and loss of taste is very unique to COVID-19. Now, there has been studies where they showed that anosmia occurs to the tune of 20 to 30% patient. There has been one study from India which has been published very recently, where they found that only 15% of Indian patients has anosmia, and the author suggested that Indian people have anosmia less frequently than the European people. But we have to realize this is not a really controlled study. This author did not study European people as compared to Indian people simultaneously. They took only historical data which are published from Europe, frequency there and frequency in India. But this study gives a message: 15% of Indian patients of COVID-19 has anosmia. This is one data set that I wanted to show you, which was published in Lancet Gastroenterology Hepatology, where they showed that the patient with diarrhea and without diarrhea, the severity differed. Like those who had diarrhea more often had comorbidity, they more often had severe COVID-19 disease, they require ventilatory support more often, they died more often. Now, this is another study, this is rather a meta-analysis of larger number of studies on a larger population. They found that actually frequency of GI symptom among COVID-19 patients and relationship with outcome, and this was published in Indian Journal of Gastroenterology. What they found is if somebody has diarrhea, he is more likely to have severe disease. Similarly, if somebody has high alanine aminotransferase, he is more likely to have severe COVID-19. So it is not only diarrhea. If a patient of COVID-19 has high liver enzymes such as alanine aminotransferase or aspartate aminotransferase, he would have more severe COVID-19. Now we studied this issue because till now, whatever we were telling were largely retrospective data, and retrospective data has limitation. The reason is whenever you do a retrospective study, that too in hospitalized patient, the problem is that the hospitalized patient would be receiving several other drugs. They would be receiving antibiotics. Antibiotics can cause diarrhea, and therefore when you say this GI symptom is related to COVID-19, it may not be so. It may be related to drugs, particularly the antibiotic that the patient is receiving. Accordingly, what we did is in our department of microbiology, where much of the work has been done by Dr. Ghoshal. Obviously, my job was only to take the questionnaire and collect the data telephonically from those patients and then analyze the data and write this. Now, what we found is that till May 17, like SGPGI started the COVID diagnostic lab in around March end. Now, till May 17, they actually tested 16,317 subjects for coronavirus or SARS-CoV-2 RNA. Of these 16,317 subjects, 252 were positive. So this sort of makes our study subject. Now, of these 252, 208 were asymptomatic. That means almost 83% were asymptomatic. Mild to moderate disease, which means upper or lower respiratory symptom, but no requirement of oxygen was there in 33. Severe means oxygen was needed in 8, and critical means needed ventilator was 5. So 44 patients were asymptomatic, and remaining 208 patients were asymptomatic. One thing to note here is most of these patients are community sample, because in India there is a system through integrated disease surveillance program of Ministry of Government of India, Ministry of Health and Family Welfare. The district sample come to SGPGI for testing. So much of these 16,000 samples are not SGPGI samples. These are all samples from the community, and here is the map of Uttar Pradesh, which gives the number of people who have been tested from different locations, and you can clearly see that the distribution is quite homogeneous in the whole Uttar Pradesh. We found 44 symptomatic patients. 18 had non-GI symptom only. 18 had non-GI symptom only. 15 had combined GI and non-GI symptom, and 11, that is 25%, had GI symptom only. So a group, rather majority, had non-GI symptom, which means respiratory symptom. 34% had GI plus non-GI symptom, and 25% had GI symptom only. What is the clinical importance of this? Today, if somebody has diarrhea, little bit fever, do not think that this is acute gastroenteritis and give him antibiotic. But what you do is get his SARS-CoV-2 RNA tested in nasopharyngeal swab. Now, look at our data. This is the pictorial summary. 252 patients from Uttar Pradesh. Now, of these 252 patients, 44 are symptomatic. Of these 44, 41% had non-GI symptom, 34% had non-GI plus GI symptom, and the remaining 25% had GI symptom alone. 33% had moderate, mild to moderate disease. Severe disease was in 8 and critical disease in 5. Now, both univariate and multivariate analysis showed that if there is GI symptom, it is associated with more severe illness. Now, this is important. Till now, whatever world literature is there, these are all retrospective study, and most of these studies, except one study from US, did not do multivariate analysis. This study did multivariate analysis and showed GI symptom is an independent predictor of severe illness on multivariate analysis. Another beauty of this study is that most of these patients are from community, and therefore they are not on multiple drug, including antibiotic, that can be a confounder causing GI symptom. Second thing, in this study, we wanted to see, are there factors which are associated with GI symptom? Yes, there are. One is comorbid illness if present, then the people are more likely to have GI symptom. If the fellow has another non-GI symptom like respiratory symptom, he is more likely to have GI symptom. And third thing is, if the fellow does not have history of contact, he is more likely to have GI symptom. What is the biological explanation for that? Biological explanation is very simple. If somebody has history of contact, you will actively test them even in absence of symptom, so that an asymptomatic patient will be picked up more easily. Therefore, he will not have either respiratory or GI symptom. On the other hand, if somebody has symptom, you are more likely to test him for SARS-CoV-2 RNA in absence of contact. Now, these are the data, multivariate analysis and univariate analysis. Another interesting thing that emerged from this study is this. So what happens is, WHO once upon a time, albeit fallaciously, said that if somebody is asymptomatic, he is less likely to transmit the virus. This is wrong. You know why? We looked at people who are asymptomatic and those who are symptomatic. We looked at people who have GI symptom, meaning they may be having severe illness as shown in this study, and those who did not have GI symptom. And we looked at cycle threshold in reverse transcripted polymerase chain reaction. What is cycle threshold? Cycle threshold means when you are doing RT PCR, you have to amplify and amplify and amplify to get the virus positive. If you have 10 RNA at least, 10 copies of RNA at least in nasopharyngeal sample, then you will be able to pick up. But if somebody has much more than 10, in lesser amplification, you will pick it up. So cycle threshold means how many times did you amplify before you got a positive result. So if somebody had to undergo less number of amplification, that is less number of cycle threshold, and you got it positive, means he has more RNA in the nasopharyngeal sample. Whereas if you had to amplify more and more, for example, 30 samples, 35 cycles or 35 cycles, that means this fellow has less RNA. And what we found is people who had GI symptom or did not have GI symptom, cycle threshold is comparable. People who are asymptomatic, people who are symptomatic, cycle threshold is comparable. What does it mean? That even the asymptomatic people without GI symptom, they have good viral load in their nasopharynx, therefore they should be able to transmit. Only thing is they are not mounting a cytokine response because they have a good T regulatory response, so that inflammation is not happening. So one more finding in our study is, out of 252 people, Fortunately, God grace, only 5 died. So all these 5, 4 of them had actually comorbidities, and these comorbidities were really potentially lethal comorbidities. So what we found is, fortunately, mortality due to SARS-CoV-2 infection in India is less compared to US or Italy. Because if you look at the pool data of US, which is available now in net, you will find their mortality rate varies from 4 to 6%. Whereas look at Indian data, mortality rate varies between 2 to 2.5%. And when you talk about a huge number, and within this huge number, there's a double the mortality in US than in India, means something, that mortality is really less. Why is mortality less in India? We have given hypothesis for that. One hypothesis is the SARS-CoV-2 RNA virus having different genotype in India? Yes, it is. In fact, we collaborated with CDRI. We have done sequencing of SARS-CoV-2 RNA from a microbiology department laboratory. Dr. Ujjwala is the primary person. And then what we did is they did the sequencing, and they found there are several different sequences in the strain that we have than what is there in US, and this has already been submitted in the electronic database. Second is, what we found is, we all know young age has less mortality. Indian population, because of survival of the people being less than in many countries, obviously is younger and they have less comorbidity. Third thing is, Indians are exposed to bad hygiene very often. From the childhood, they live in a bad hygiene. And we know that if hygiene is too good, it leads to more allergy, it leads to inappropriate cytokine response, it leads to more autoimmune disease. So this is called hygiene hypothesis. Maybe because of the poor hygiene, India has better T regulatory response, and that's why they are not mounting so much cytokine response. I know some author published that in India, BCG vaccination in the childhood might be one of the reasons for less mortality, but I don't believe that. I would say BCG vaccination is a surrogate marker of high infective load, and high infective load is associated with good T regulatory response. There is one study from Hong Kong which showed that gut microbiota in India, gut microbiota decide how severe coronavirus disease 19 would be. And this is quite plausible. The reason is very simple. We know that gut microbiota actually manipulate or modulate the immune response of the body, the whole body, not only the gut. And therefore, I would believe that India having less hygiene may have different gut microbiota. This has been already shown. Also, a large number of people are vegetarian. Vegetarians have different microbiota, and therefore this may be one of the explanation, and these are the explanation that we have proposed. Now, can SARS-CoV-2 RNA be there in other sample, like stool sample, like urine sample, like blood sample, like salivary sample, and so on, bronchoalveolar lavage fluid sample? This is a study from China where they showed, yes, in a large number of body fluid and body tissues, SARS-CoV-2 RNA would be there, but among these, urine sample is least often having SARS-CoV-2 RNA. This is the meta-analysis of all the paper, which showed that actually almost 50% of SARS-CoV-2 coronavirus disease 19 patients have SARS-CoV-2 RNA in their stool, and those who have GI symptom more likely to have SARS-CoV-2 RNA in their stool than those who have no GI symptom. Also, those who have diarrhea have higher viral load in the feces than those who don't have diarrhea. This is one paper from China where they did endoscopy. We know doing endoscopy in coronavirus disease 19 patient is very risky, because during endoscopy, patient actually have a lot of gag, and this gag generates a lot of aerosol, and therefore the doctor doing endoscopy will be at high risk. And this is one published gut paper where they did 1 or 2 patient endoscopy, and they found on endoscopy you can find mucosal damage in COVID-19 patient suggesting maybe it has invasive potential in the gut. Now, this is the temporal pattern of RNA excretion, and let me tell you that the RNA of the virus can be there in the stool almost 2 weeks after it has become negative in the nasal swab or oral swab. This has an epidemiological connotation. The reason is that maybe like polio, SARS-CoV can lead to community transmission through the fecal-oral route because it is lasting in fishes for long. Can you find the RNA in sewage water? These are some of the studies from the West where they found even the sewage water can carry SARS-CoV-2 RNA. And this is one study from Hyderabad, India, where they showed sewage water to have SARS-CoV-2 RNA. Now, what is the impact of this pandemic or epidemic in our country on GI healthcare services? We did a all India survey among the gastroenterologists and endoscopists. We wanted to know how much is gastroenterology and endoscopy care for the patient being affected by this COVID-19 pandemic. And this survey was primarily done from Apollo Calcutta with collaboration from us. And what we found is routine endoscopy services actually got interrupted or got reduced to 10%. So whatever endoscopy services was being given earlier before this epidemic, the endoscopy services has come down by to 10%. That means reduction has been to the tune of 90%. So you can imagine how much chaos this virus has caused, because all those who needed these services actually are not getting these services, and these diseases were also potentially fatal. So therefore the life loss is not only happening because of the COVID-19, but due to other reasons which is affecting the healthcare. Now, how to step up the healthcare? This is one of the international review that we did with Australian, with Hong Kong, Chinese, and Singaporean group. And what we did is we tried to find out how can we give the services, because obviously if we have tremendous attack of COVID-19 among healthcare professionals, it will be another great chaos, because till now, at least if you can save your healthcare professional, they are like your army member. You can win the war, because this is like, if my army is exhausted, then any war we have to lose. But if my army is somewhat intact, then maybe we can win the healthcare. And this is the method. The method is telemedicine. And I must acknowledge the tremendous contribution of telemedicine, School of Telemedicine and Biomedical Informatics, that was established by Professor S.K. Misra from the scratch in last 25 years in SGPGI, because we were prepared much before. Like this article talks about all this preparation. So many countries had to prepare all this, but at least fortunately in SGPGI, we were prepared much before the courtesy or all the credit goes to School of Telemedicine and Biomedical Informatics. So these we are seeing the patient. This is a tele-OPD. So typically we use a sort of a video conferencing system like WhatsApp. I use this website through which patient can take appointment. I can schedule his appointment. I can write the advice form. I will not say prescription, because fortunately now telemedicine has become legal. I wrote it as advice or advice earlier, because I was afraid to write prescription there because of the litigation. And patient can use a password and username, click on this pen and paper, and can download the advice that is given through this system. Until now, we have seen huge number of patients through telemedicine services. Therefore, I conclude that GI symptoms are not uncommon in patients with COVID-19. Presence of GI symptom is associated with severe disease. Fecal viral excretion is reported in 50% patients with COVID-19, and fecal viral excretion may continue even after nasopharyngeal virus has become negative. Therefore, those who unfortunately got COVID-19 and now on follow-up their RNA has become negative in the nasopharyngeal swab, don't be too happy. Don't think you are absolutely safe for your near and dear ones. You can still transmit them through fecal-oral transmission. Keep precautions so that you use separate toilet, do disinfection of the toilet after you have gone to the toilet. And SARS-CoV-2 should be tested in feces, because we gastroenterologists used to treat several diseases with fecal transplantation. In today's time, if I have to do fecal transplantation, I'll be worried. One. Second is, if I have to do fecal transplantation, I will test the fecal material again and again for SARS-CoV-2 RNA before transplanting to another person. Thank you so much.